So the first activity that we're going to will be tackling for bacteriology laboratory is the control of microorganisms, specifically disinfection, sterilization, and microbiology safety. Now you're familiar, class, that the subject is bacteriology. This involves the study of bacteria. And as you're all aware, bacteria is capable of causing disease. Since we are medical technologists, or you will be future medical technologists, it is very imperative or very important, class, that you need to know, or there is a must to know for you how to handle or how to remove or how to kill or properly dispose of bacteria. So in this first activity, you will learn how to control microorganisms, how they are disposed, and how would you protect yourselves against them. Now let's talk about the different methods of disinfection and sterilization. Now the first method, class, are known as physical methods. Now when you say physical methods, you are using agents without the use of any chemicals. Meaning, class, gumagamit ka ng agents or substances that would kill bacteria without any chemicals. Now, this would include the following. The most common, class, is moist heat via autoclaving. So this is the most common and effective method for sterilization. This has the principle. So the principle here, pag tinanong sa quiz nyo, what is the principle of autoclave? Pakitandaan, steam under pressure. So there is steam. When you say steam, there is heat and there is pressure. Usually 121 degrees centigrade for 15 minutes. And it would be able to denature protein and kills both vegetative cells and spores. So let me repeat, class. The first physical method is moist heat, also known as autoclaving. Most common method, the principle is steam under pressure, usually at 121 degrees centigrade for 15 minutes. Paki-include ito at 15 PSI. PSI, class, refers to the unit of pressure. And if asked how does it kill, pag tinanong how does it kill your bacteria, it would denature protein, killing both vegetative cells and spores. Another physical method, class, is dry heat. Now, for dry heat, you would use higher temperature. Kung sa moist heat, 121, sa dry heat, higher temp. Usually 160 to 170 degrees and a longer amount of time, 2 hours. Suitable for materials that may be damaged by moisture. So this is not this is recommended, class, for equipments, materials that are not allowed to be wet. Hindi pwedeng mabasa. So you're going to use dry heat method for materials that are not allowed to be wet. It kills by oxidative damage and protein denaturation. Then another method, class, is filtration. Now this method would physically remove microorganism from heat sensitive fluids using membrane filters with pore sizes typically 0.22 micrometer. It is not bactericidal but is effective at removing fungi and bacteria. So sa filtration, class, the thing you need to take note here is that it does not kill your bacteria or fungi. What it does, class, is that it would just remove them from fluids by using filters with a pore size of 0.22, 0.22 micrometer. So that's the third one, filtration. So again, filtration does not kill the bacteria. It just removes them from fluids because it uses a pore size, membrane filters with pore sizes at a size of 0.22 micrometer. Then the fourth physical method is radiation class. Now, when it comes to radiation, it uses ultraviolet light, or also known as your UV light. Now, UV light would be directed against the organism, the microorganism, causing DNA damage. It would destroy, class. It would destroy thymine dimers and is used for surface sterilization. Ang ginagamit, class, the ultraviolet light used here is ionizing radiation, specifically gamma rays, for sterilizing disposable medical supplies. Let's go to the second method, the chemical methods class. So we're done with the physical methods. Pag physical method, again, it does not use any chemicals or substances. Usually heat lang. But for chemical methods class, you're going to use what we call disinfectants and antiseptics. Now, chemical disinfectants and sterilants are used for surfaces, instruments, and in some cases, the skin, depending on their toxicity and spectrum of activity. Now, when it comes to chemical methods class, there are two words here that you need to identify or differentiate. Si disinfectant and si antiseptic. Ang tatandaan nyo lang class, kapag si disinfectant, they are usually used sa surface. Kapag antiseptic, they are used sa balat or the skin, specifically living tissue. The most common type of disinfectant class is your Sunrox, while the most common antiseptic is your alcohol. So Sunrox or bleach class is the most common disinfectant. Antiseptic is alcohol, specifically 70% alcohol. So that's the difference between disinfectant and antiseptic. Kapag disinfectant, inanimate surfaces. Pag antiseptic, use on skin or living tissue. Now, let's talk about the common chemical agents. The first one, class, is the alcohol. Now, the alcohol, familiar kayo dito, is the one that's commonly used is ethanol. 70% ethanol and 70% isopropanol. Now, they would act by denaturing proteins and disrupting membrane. They are rapidly bactericidal but not sporicidal. Pag sinabi mong sporicidal class, there are certain bacterias, please take note of this, there are certain bacterias that contain what we call spores. Now, itong mga spores na to, class, this allows the bacteria to live in harsh environment. So kapag ang bacteria mo may spores, class, they are able to survive in very harsh and in very deadly climate in areas such as the surface or the ground. Now, the problem with alcohol, if you have this type of bacteria class, they are not able to destroy the spores, but they are bactericidal. They can easily kill common bacteria. But when it comes to spore-bearing, mga merong spore na bacteria, it's very difficult to kill them. Now, alcohol class are usually used as skin antiseptics and for small surface disinfection. So, sa laboratory nyo, you will be using this type of disinfection alcohol para sa surface. No need to bring sunrock siguro panglinis sa lamesa, just the alcohol. And the most common na pinanglilinis nyo sa kamay nyo aside from soap and water is of course skin is alcohol. Now the second common agent, chemical agent class is aldehyde. The best example is glutaraldehyde. This is a high-level disinfectant and chemical sterilant. It would alkalite nucleic acid and protein. Very effective siya class for spore-bearing bacteria upon prolonged exposure. So when it comes to killing spore-bearing bacteria, the best method would be aldehyde, used for heat-sensitive instruments like endoscope. The third agent, chemical agent are halogens class. Now the best example of halogen is yung iodine nyo. So familiar kayo sa iodine, familiar kayo sa chlorine. Iodine is an effective antiseptic often combined with povidone. Yan yung tinatawag nilang betadine class. So iodine with povidone is usually called also as your betadine. They function by oxidizing cell components and inactivating enzymes. Then another one class is your chlorine compound, specifically yung sodium hypochlorite nyo or yung bleach or yung brand name na Sonrox. They act by oxidizing cellular components effective for blood spills. So usually class pananglilinis siya sa mga spillage. Highly effective siya for spillage. Take note, class, that there is a required 1:10 dilution of bleach for disinfection of surface and lab. Pag sinabi mong 1:10 dilution of bleach, we refer to this as 10% bleach concentration or 10% sodium hypochlorite. So whenever you would clean the surface using Sonrox using sodium hypochlorite, you need to dilute it at a 10%. So how would you do that, class? Let's say you want to prepare 100 ml of 10% sodium hypochlorite. Basically, you would use here 90 ml water and 10 ml concentrated Sonrox or bleach. So again, if you want to prepare 100 ml of 10% sodium hypochlorite, you mix 90 ml water and 10 ml sodium hypochlorite. Then we have your detergents, also known as your quartz, quaternary ammonium compounds. Now, these are cationic detergents that would disrupt membrane and denature protein. So, itong mga to, class, itong detergents na to, these are positive ion substances. Now, whenever they would enter or they would get attached to the surface of bacteria, because of this positive charge, they would destroy or they would disrupt the surface or the covering of your bacteria. This is very effective against gram-positive bacteria and envelope viruses. Used for general surface cleaning but ineffective against spores and some gram-negative bacteria. Then we also have your phenolics derived from carbolic acid. They act by disrupting cell wall and precipitating protein. Effective against gram-positive bacteria and your mycobacteria. Ang pinaka-common example class ng phenols nyo is yung Lysol na spray. By using the Lysol na may spray glass, it contains phenol. So, pwede siya pang use sa floors and even countertops. Then another one are heavy metals. Less commonly used today due to toxicity. So rarely use ang heavy metals class for disinfection. A most common example is silver nitrate. It would act by precipitating protein and interfering with metabolic processes. Before, it was used in eye drops class for newborns against Neisseria gonorrhea. Then gases will also be used. Example is ethylene oxide, used for sterilizing heat-sensitive medical equipment. Acts by alkylating DNA and protein, requires aeration after sterilization due to toxicity and flammability. Now let's talk about surgical hand scrub and waterless surgical hand rubs class. So usually class ang tao, if ever a person is in a rush, most of the time ang tao, they're always in a rush that they tend to forget to wash their hands. Now, to remedy this, to somehow solve this, gumagamit sila ng what we call sanitizers. Yung mga gel sanitizers, alcohol sanitizers. Now, there's a very big difference between washing your hand using soap and water and washing your hands using these sanitizers. Now, if you're going to wash using soap and water, you need to do what we call a surgical hand scrub. This is the procedure done by surgeons prior to doing a surgery. So this is also applicable to us class. Pwede rin natin itong gawin. Now this would use antimicrobial soap. Usually chlorhexidine or povidone iodine. But in our case, we can just use regular soap, pwedeng safeguard. Now, in doing this, in doing a normal washing of hands using soap and water, there is a proper step-by-step procedure that needs to be followed from scrubbing the fingertips to the elbows, and there would be a specific duration, meaning my oras class, there is a specific time. It doesn't have to be 10 seconds. It's usually 2 to 6 minutes. It's that long to ensure that there is the proper removal of debris and microorganism. So that's why class, between using sanitizers and washing your hand using soap and water, mas effective pa rin talaga maglinis ka ng kamay using soap and water. You basically remove everything, every possible contaminant from bacteria, from any dirty debris. While waterless surgical hand rubs naman class are usually alcohol-based rubs, either containing ethanol, isopropanol, or even N-propanol. Now, this is time-saving and an effective alternative to scrubbing. They would often contain emollients class to protect the skin. Now, pag sinabi mong emollients, these are substances that would hydrate the skin. So, para siyang moisturizer. It acts as a moisturizer class. Effective to apply to clean, dry hands before gloving. Although they do not physically remove debris, they are rapid, broad-spectrum agents against bacteria and viruses. So, that's the difference between a hand scrub and a waterless hand rub. Now, let's talk about the pre-surgical skin disinfection. Now, as medical technologies class, it's very important that you know this because in our profession, there's what we call a blood, a test known as your blood culture and sensitivity. Now, sa blood culture and sensitivity, you are required to collect blood in a aseptic manner. Pag sinabi mong aseptic manner, yung letter A dyan na prefix, yung A na prefix dyan means absence of anything septic or dirty. So you need to make sure that when you collect the blood, the site of entry, the area where you would be collecting the blood or where you be inserting the needle, has to be properly cleaned. Now, aims to reduce microbial flora at the site of collection or the incision site, thus minimizing the risk of post-operative infections. Now, ang ginagawa dito, class, you would use povidone iodine or in some case, chlorhexidine gluconate. You apply it in a surgical motion outward from the surgical site. So, kung let's say ito yung arm, you start here. This is your collection site. Dito ka mag start Then, you go in an outward manner using the povidone iodine. And then after that, so tatlong step yan, class, to give you an idea. Paki-take note na lang. First, you clean it using your povidone iodine or your betadine. Then, rinse it with alcohol. Then, let it air dry. So, betadine, then alcohol. Now, you may be wondering, sir, bakit may alcohol? The purpose of alcohol here is to remove the stain of the betadine. So, pag tinanggal mo yung betadine, mas presentable, mas malinis tingnan yung site of incision mo. Then, you let it air dry before collecting the blood. Now, very effective itong povidone iodine class and chlorhexidine gluconate, and they would exhibit broad-spectrum antimicrobial activity. When you say broad spectrum, from the word broad, meaning many, they are effective against many bacteria and even viruses and fungi on intact skin. The disinfectant is allowed to dry to maximize effect and minimize chemical irritation or fire hazard during surgery or collection. So that's how pre-surgical skin disinfection is applicable sa atin, class, in the test blood culture and sensitivity. Now, let's talk about the safety program for the clinical laboratory. Now, as medical technologies class, you need to know the safety precautions that are applied sa laboratory. Now, there is what we call a universal safety protocol to all patient care settings, assuming that all blood and body fluids are potentially infectious, class. So whenever you would handle sample in any laboratory or in any of your laboratory subject, whether it is from your classmate, whether you collected it from your relative or not, you need to consider it as potentially infectious. Let's say nag-collect ka ng blood sa boyfriend mo, sa girlfriend mo, you still have to consider that as potentially infectious. You collected sputum from your relatives, from your father, mother, you still have to consider that as potentially infectious. Thus, you need to apply universal safety protocol. So again, class, regardless of where the sample came from, you have to consider them all as potentially infectious, thus applying universal safety protocol. Now, ano ba yung mga universal safety protocol? The first one would be hand hygiene, how you wash your hands. The second one would be the use of your PPE. The third one is the safe injection practices. Fourth one is respiratory hygiene and cough etiquette. And the fifth one would be proper handling of patient equipment and laundry. So these are the requirements, class, specifically dito sa pag-use ng PPE nyo. So there's a proper order. There's a proper order of wearing or donning, yung pagsusuot, and pagdodoffing or pag-remove, which later I will explain. Now, they have what they call an exposure control plan. Now, this would outline procedures to minimize occupational exposure to bloodborne pathogen. So ito yung mga ini-implement class ng isang hospital to protect their employees, their healthcare workers against these diseases, these bloodborne diseases such as your HIV, hepatitis B, hepatitis C viruses. They would include risk identification, hepatitis B vaccination availability, post-exposure evaluation and follow-up, documentation and training of employees under OSHA's bloodborne pathogen standard. Then there are also what we call another program class are transmission-based precaution. So these are additional measures used for patients with known or suspected infections that spread via contact, droplet, or airborne routes. To give you a story class, in some hospitals, if they identify a patient that has TB or tuberculosis, the nurses are obliged to notify the other sections. They need to notify the laboratory, the radiology, and other departments that this room, that this patient has tuberculosis. The reason for that is so that the healthcare workers that would visit the patient would wear the necessary or appropriate PPE. So ito yung mga transmission-based precaution. They are used for patients class that would spread the disease via contact, droplet, or airborne routes or airborne routes, sorry. So they would usually involve the use of gloves and gown, surgical masks within 3 feet for droplet precaution, then for airborne precaution class N95 respirator and negative pressure rooms for TB patients. So for contact precaution, gloves or gowns, pag droplet, surgical mask within 3 feet, airborne precaution, N95, and negative pressure rooms. Then there are what we call engineering controls class. Now, when it comes to engineering controls, it would involve devices or technology. The most common example are your biological safety cabinets, 1 to 3. Needleless systems. So there are systems in collecting blood that would lessen the exposure to needle to the healthcare worker. Sharps disposal container, mechanical pipetting devices, they are all considered first line barriers in lab safety. Then there's also your work practice controls. This refers to behavior or procedures that would reduce exposure risks, such as eating, drinking, or applying cosmetics in the lab area. So pag naging intern na kayo class, you're not allowed to eat, you're not allowed to drink, you're not allowed to wear makeup in the lab sections. Then hand washing always after removing gloves, proper decontamination of work surfaces, minimizing splashing or spraying during specimen handling. Now, let's talk about PPE naman class. So, these are worn. Itong mga personal protective equipment na to, they are worn to protect lab worker from exposure to infectious materials. Usually, they are worn class depending on the risk of exposure, the nature of the procedure, and the biosafety level. So again, PPE are selected in use depending on three things: risk of exposure, nature of the procedure, and biosafety level. So PPE class would include gloves, gowns or lab coats, face shield, masks, and eye protection, specifically goggles. PPE class must be readily accessible, properly fitted, inspected before use, and appropriately disposed or disinfected after usage. Now let's talk about the donning and doffing. So how would you wear your PPE. Now, there is a standard sequence. So, as I mentioned earlier, when you wear your PPE, there is an order or a proper order of wearing them. So, you would wear them normally before entering the laboratory or beginning any procedure. You start first class by wearing your gown or lab coat. Fully covered torso from neck to knees, arms to wrists, and wrap around the back. Fasten at the neck and the waist. Then the second PPE to wear would be your mask or respirator. So you need to make sure that the ties or bands are properly secured. Flexible band to the nose bridge and snug to face and below the chin. Then goggles or face shield. Gloves class would have to extend until the wrist. So dapat aabot siya sa wrist class and would overlap with the gown sleeve. Then before donning or wearing your PPE, always perform hand hygiene. And then you could contact to prevent contact with any potentially infectious material. Now for doffing naman class, usually done removing the PPE is done before exiting the laboratory or after completing a procedure. Kung sa donning, ang first na sinusuot nyo is the gown. Sa doffing naman, ang first na tinatanggal nyo is the gloves. They are removed first, being careful not to touch the skin. Use a glove-in, glove technique to avoid contamination. Followed by goggles or face shield, gown, mask or respirator, and then end it by performing hand hygiene. Perform immediately after removing all PP. So that's the step-by-step procedure on donning and doffing. So kindly memorize the step class. They may come out in your quiz. Now let's go to the biological risk assessment. So this is a process class to identify what are the potential hazards associated in handling biological agent. So itong assessment na to class, it helps you prepare. It helps you prepare on how you would be handling a certain procedure or handling a certain microorganism. And to help you identify, they develop what we call biosafety levels. Now, there are four biosafety levels. Let's tackle them one by one later. So, let's first go to the factors used in assessing them. The first one would be pathogenicity and transmissibility of the agent. Now, when you say transmissibility and pathogenicity, this refers to the ability of the microorganism to cause disease. How does it cause disease class? Gaano ba siya kabilis magkalat ng sakit? Now, this is also the potential of the agent to cause illness. Transmissibility naman class would assess it whether it would spread through air, contact, droplets, or vectors. Example nito class is yung E. coli. It has a very low pathogenicity. Since low ang pathogenicity niya, low lang yung chance niya mag-cause ng sakit, it undergoes or it belongs to biosafety level 1. But if it's your Mycobacterium tuberculosis that is known to cause your TB, airborne transmission siya class, highly pathogenic, meaning napakadali niya mag-cause ng sakit, biosafety level 3. Then for Ebola virus, highly pathogenic na, highly transmissible pa by body fluids. Therefore, it belongs to biosafety level 4. So the greater the ability to cause a disease, the easier for it to transmit, the higher the biosafety level needed. The second one class is the procedure involved. Now, these are factors that would involve manipulations which would generate infectious aerosol. So, yung mga procedure ba na ginagawa mo class would produce infectious aerosols or increase exposure risk. Ang mga example nito class is vortexing or centrifuging sample. pipetting infectious fluid, culturing bacteria in open system, performing necropsies or specimen alicoating. For example, class, if nag-work ka sa school ng Staph aureus, you may only use biosafety level 2 precautions. But if you're using sample, nag-collect ka ng sputum from someone na may tuberculosis, then you are required to apply biosafety level 3 controls. Then the last one is the immune status of the personnel. That's why class important bago kayo mag-intern, you need to have a complete set of vaccines, specifically for your hepatitis B and such. So this would identify, or they would usually involve mga immunocompromised, pregnant, or vaccinated against the agent. The goal here, class, is to match task assignments and controls with individual health risks. Example, class, if a person is vaccinated with hepatitis B, they may safely handle bloodborne pathogens such as HIV, hepatitis B, hepatitis C. Then a pregnant worker may be restricted from handling listeria, which poses a higher risk to fetal health. Then immunocompromised staff may require reassignment to lower risk tasks even within the same biosafety level facility. So kung yung person immunocompromised, mahina yung immune system niya, expect mo siya class that he would be given lower risk jobs or tasks. Then processing of patient specimens. So whenever you would handle, please take note of this class, whenever you would handle specimens in all of your laboratory classes, you need to wear gloves. Then transport specimens in a sealed, leak-proof container. So pag nag-collect kayo ng blood, make sure that it is properly sealed. Then decontaminate surfaces after spills, avoiding aerosol generation. Then all specimens should be treated as potentially infectious, especially blood, CSF, and respiratory fluids. So let's go naman class to your biosafety cabinet. So ang biosafety cabinet nyo class, these are the one of the engineering controls. These are primary containment devices that would have three purpose class. It would protect the worker, the environment, and the specimen. So it protects the worker from exposure. So pinoprotektahan niya tayo class from being exposed to the bacteria. Then the environmental from accidental release of the pathogen. So an example class yung mga movies na nakikita niyo, yung mga zombie movies, yung 28 days later, 28 weeks, 28 years later. So those are hypothetical situations wherein the microorganism is released to the environment causing a massive problem. So by using a biosafety cabinet, you would be able to somehow prevent that. And the next one is the specimen from being contaminated. So it's very important, class, that you would isolate your target organism. Pag sinabi mo ang target mo lang is Mycobacterium tuberculosis, your only target should be MTB. So there should be no other bacteria. So by using a biosafety cabinet, you allow a lesser possibility for contamination. Now there are three classes for BSC, class 1, class 2, and class 3. Sa class 1 class, it offers personnel and environmental protection but no product protection. Meaning, class, you're not able to protect your sample from contamination. Air is drawn inward and filtered before being exhausted. While for your class 2 BSC, divided into four types, A1, A2, B1, and B2, it provides protection for personnel, environment, and product. So sa laboratory class, sila yung pinaka-commonly used for handling MTB. Then there's also a HEPA filter, both before reaching the work surface and before being exhausted. And the last type, yung Class 3 Biosafety Cabinet, also known as the glove box. This provides the highest level of containment, fully enclosed and gas tight. Usually, ganito itsura niyang class. May salamin dito. So this is an entire glass. Tapos dito sa ano, may dalawang butas. That is where you would insert your hands. Then you would start manipulating the sample without any direct. So yung kamay nyo lang yung nakaloob doon class, and it's is in a big glove, something like that. Usually used for biosafety level for agents such as your Ebola viruses. So those are the three biosafety cabinets: Class 1, Class 2, and Class 3. Now let's go to the biosafety levels 1 to 4. So ito yung dinesign nila class on how you would apply different PPE, different protocols, different procedures. Now let's go to biosafety level 1. Now biosafety level 1 would cover agents that are well characterized, meaning matagal na silang identified class to not cause diseases. So hindi sila nagko-cause ng disease in healthy humans. An example of these organisms class are Bacillus subtilis, Lactobacillus acidophilus, Micrococcus luteus, Escherichia coli, Corynebacterium cirrhosis. For fungi, we have your Saccharomyces cerevisiae, also known as your Baker's yeast, Penicillium notatum, non-toxigenic strain. And for the practices sa BSL-1 class, standard microbiological procedure, lab coat, gloves as needed, as well as eye protection. Then facility requirement, open bench work. So if you've already seen the tabletop sa GDLSC, that is as example of an open bench work. So lamesa lang talaga siya. Then you have your sink and easily clean surfaces. No special primary or secondary barrier. Usually example ng mga BSL-1 na procedure class sa mga teaching lab and even in university. Excuse me again. Let's go to the second one, yung biosafety level 2 naman. For biosafety level 2 class, this is a moderate risk pathogen associated with human disease, specifically Staphylococcus aureus, Salmonella. So ito yung mga involved class: S. aureus, Salmonella enterica, Shigella, E. coli, the pathogenic strain, Clostridium difficile, Haemophilus influenzae, Neisseria gonorrhea, Listeria monocytogenes, Yersinia enterocolitica. Then viruses can also be included here class: hepatitis B, hepatitis C, HIV, influenza A, herpes simplex virus. Then fungi, Candida albicans, Cryptococcus, and Aspergillus fumigatus. So you would apply the practices in BSL-1 class plus limited access, biohazard signage, sharp precaution, biosafety manual. Then you're required to wear lab coat, gloves, face protection. Facility requirements are self-closing door, autoclave should be available, sink with eye wash. Then you also need to use a containment device, specifically Class 2 biosafety cabinet, to prevent aerosols from being released class. Example using biosafety level 2 are diagnostic clinical laboratories. So most laboratories class in hospitals are following the precautions in a biosafety level 2. Now let's go to the more deadlier or more dangerous one class, yung biosafety level 3. Pag biosafety level 3 class, ang tatandaan mo lang dyan, they are usually transmitted or they are able to transmit the disease via aerosol. and they could be severe or potentially lethal, nakakamatay. Example are your TB, Mycobacterium tuberculosis, Francisella tularensis, Brucella species, Coxiella burnetii, Burkholderia pseudomallei. Viruses such as your SARS-CoV, MERS-CoV, yellow fever, Venezuelan equine encephalitis virus, chikungunya virus, and even your COVID class. Your COVID belongs also to biosafety level 3. Fungi, Coccidioides immitis, and Histoplasma capsulatum. For biosafety level 3 class, very strict on access. So only a certain limited individual are allowed to handle BSL-3. Then the contamination of all waste and lab clothing before laundry. Then you also have to collect baseline serum collection. So you need to identify if your facility workers are in a healthy status. Then scrubs are required, front gowns, double gloves, and N95 respirator. Then for facility requirements, we have your negative airflow, sealed windows, HEPA filtration of exhaust air. Then all manipulations class should be performed in a class 2 or class 3 by safety cabinet. So pag magvo-vortex ka, if you're gonna vortex, if you're gonna centrifuge, it has to be done inside a biosafety cabinet. Example of these are TB reference lab and some research facility. And the highest level class designed for dangerous and exotic agents with high risk of aerosol transmission and life-threatening disease with no available vaccine or therapy. So walang treatment class transmitted via aerosol. Example of these are your Ebola, Marburg, Lassa fever, Crimean Congo hemorrhagic fever, Nipah virus, Machupo virus, Rift Valley fever. So pag level 4 na class, mostly ang kasali dyan are viruses. So you need to have a complete clothing change before entry. Shower on exit. So maliligo ka talaga class after performing the procedure. Usually dito ang suot mo para kang naka astronaut na attire or para kang naka diving suit, fully enclosed class. You wear a full body, air supplied, positive pressure suit, or you would use class 3 biosafety cabinet. So full body ka class, may sarili kang air supply. Then isolated zone, so far from residential area or commercial area. Then it has its own supply and exhaust system, vacuum system, and decontamination system. Class 3 BSC or full body suit in dedicated building. CDC and maximum containment units in specialized research facility. So far class ang dito sa Pilipinas, we only have up to biosafety levels 1 to 3. Wala pa tayong biosafety level 4 in our country. Now let's talk about the different hazardous waste in the clinical lab. So they would refer to substances that would pose risk to human health. So iba-iba yung mga hazardous waste class. They could be biological or chemical hazards. So let's talk about them one by one. So we have your infectious or biohazard waste. This would usually involve human blood, body fluids, used swabs, gloves, pipettes, discarded specimens from patients suspected of having infectious diseases. So usually kapag infectious or biohazard waste class, they are to be disposed in a yellow trash bag. Kapag naman sharps waste, designed for needles, scalpel, lancet, must be disposed in a puncture-resistant container labeled with a biohazard symbol. So kapag sharp naman class, you have there your puncture-resistant container Then you would dispose it in a red trash bag. Then we also have your chemical waste. These are unused reagents, staining solutions, cleaning agents that may be flammable, corrosive, or toxic. Volatile solvents such as xylene, ethanol, and formalin. Then we have your pharmaceutical waste, mga expired na antibiotics, medication. Then radioactive waste, less common in routine microbiological lab. This would include radioactive isotopes used in tracer study. So kapag radioactive waste naman class, orange trash bin. So pakitandaan yun. So proper disposal class is essential. So usually in disposing waste, you would use different methods. The first and the most common is autoclaving. So pwede mong i-autoclave yung waste mo class bago itapon. So this is used for decontaminating cultures, plates, and infectious liquid waste. Then another one is via incineration, used for solid biohazardous waste that cannot be autoclaved such as your gauze or your PPE. So usually ini-incinerate sila kasi sinusunog sila. Then chemical disinfection, so liquids may be disinfected using a 1 is to 10 sodium hypochlorite solution. Then segregation class and place in color-coded containers. So yun nga yung sinasabi ko, if it's so in residential area, ang ginagamit natin glass are usually black and green trash bin. So kapag black are usually the non-biodegradable. Kapag green, ito yung mga biodegradable. So kapag mga non-biodegradable, hindi nabubulok such as paper, carton, something like that. Pag for green ones, these are biodegradable, food waste, etc. But when it comes to sa workplace natin, yellow would be for infectious, red would be for sharps, orange would be for radioactive. So very important class that when you dispose waste, you follow this color-coded disposal. Then labeling, all containers must be labeled with the biohazard symbol and content. Then we also have your chemical safety. So working with chemicals in a clinical lab would require awareness and compliance with protocol. So whenever you would open a new chemical class, mag-open ka ng bagong chemical, you need to check MSDS. MSDS stands for Material Safety Data Sheet. It contains the must know class, ano yung mga danger regarding the chemical, the properties, the risks, the first aid, and how to dispose it. Then proper labeling. So pag open mo ng reagent, you need to clearly label it with the name, the concentration, the preparation date, and the expiration date. Then when you would handle volatile chemicals class such as formaldehyde, phenol, you need to use a fume hood. PPE naman class when handling chemicals. Then your spill kit, let's say nagkaroon ka ng spillage. Your spill kit should be accessible and staff trained to neutralize and clean up spills safely. Let's say class natapon yung sputum ng patient. So you need to have a spill kit available to clean this up. Then signage. So proper signage is also necessary class. So you have your biohazard sign placed at entry points to BSL2, chemical hazard signs, warnings for flammable, corrosive, etc. Then emergency exit and equipment labels. Mark the location of your emergency shower, eye wash station, radiation hazard signs for labs handling radioactive material. Now let's go to the classes of fire extinguisher. So these are a must-know class. So there are different classes. Let's tackle first the first one, yung class A, or used for ordinary combustibles class. Designed to extinguish fires involving ordinary combustible materials. Example class ng mga materials na to are paper, clothing, wood. Um, Fire would usually start from paper glass. So, very important tong fire extinguisher na to. Ang agent na ginagamit ng ganitong fire extinguisher class are water and a dry chemical, usually monoammonium phosphate or foam. Use a sweeping motion at the base of the flame. So, pag class A class, again, used for fires caused by ordinary combustible material such as paper. Pag class B naman class, used for liquids and gases. Targets fires caused by flammable or combustible liquid. Example are ethanol, methanol, acetone. These chemicals are used in staining, decontamination, or preservation. So, very important class na meron kang class B na fire extinguisher. Ang extinguishing agent dito, it contains carbon dioxide, foam, or dry chemical ABC type. Never use water glass. It may cause the fire to worsen by spreading the liquid. And then we also have your Class C fire extinguisher. So kapag Class C naman, usually electrical equipment. So kapag Class A, again, ordinary combustible. Pag Class B, flammable liquid and gases. Kapag Class C, electrical equipment, involves energized electrical equipment. Example, fire from centrifuge, incubator, etc. Electrical faults are common fire risks sa lab with high-powered instruments. Since a lot of equipment class sa laboratory, they consume a lot of power. There's a possibility that it would cause an accident. Pwede siyang mag-short circuit, pwede siyang ma-overcharge causing a possible fire. Ang extinguishing agent dito is CO2 or dry chemical. Non-conductive agent must be used. And then class D naman class for usually designed naman to for combustible metals. So mga metals naman. So pag A, common combustible, B, flammable liquid and gases, C, electrical, kapag D naman, metals. Example are magnesium, titanium. Ang extinguishing agent niya are dry powder agents. So that's your class D. And the last class, yung pinaka-last na class ng fire extinguisher is the class K. Usually for cooking. So ito yung mga fire extinguisher nyo for residential class. Usually control fires from cooking oil or fats. Usually from grease fires. Not applicable sa clinical lab but could be used sa hospital or institutional kitchen. So, ang extinguishing agent dito is a wet chemical agent that would form a soapy foam to suppress flame and prevent reignition. For kitchen setting lang siya. Then, recommended extinguisher nyo class is the ABC type. Pag sinabi mong ABC type class, it's already a combination of the three. Now, use for ABC fires siya. They contain monoammonium phosphate and is effective across multiple hazard. So kapag si class ABC type na fire extinguisher class, ang meron siya is monoammonium phosphate which is very effective across multiple hazards. So this is what you will be doing. So we're done with the explanation on the activity 1. Now this is what you will be doing class in the practical. So these are the steps and it should last, paki-take note, it should last for 60 seconds. So you start by wetting your hands, apply the soap, rub hands palm to palm. So palm to palm class, both hands. So you start from your left or your right or to left. Kayo bahala where you want to start. Now you rub your palms palm to palm. Then after that, you do that for around 8 seconds. 8 to 10 seconds. Now you go to the next step. You rub the back of each hands with fingers interlace. Kapag sinabi mong interlace, you have your hands spread and you insert your hands, fingers, dun sa mga spaces in between. Kaya ganyan siya. So that's how you would rub the back of each hand with fingers interlace and you move in a back and forth motion. Once done with that class, do it again for 8 to 10 seconds. You now go to the next step. You rub your palms together with fingers interlaced. So nakaspread ulit yung mga fingers nyo. Do that again for 8 to 10 seconds. Then rub with the back of fingers with opposing palm. Then rub each thumb, clasp in opposite hand. Then rub the tips of the finger. Then lastly, last step nyo class is the wrist naman. So dito sa rubbing the tip of the finger class, this has to be done in a circular motion. Rinse with water, dry thoroughly with your hands, and your hands are now clean. So that's how you would do proper hand washing. Don't worry class, prior to the start of the practical, I will demo this to you so that you would somehow have an idea. But I still want your class to practice this. Kaya nyo naman to, you have there later, I'll show you the activity sheet that you would be using para may guide kayo. Now, you're also going to remove your gloves class. So, follow this instruction. So, you start by removing the non-dominant non-dominant glove nyo. So kung ikaw ay, ang dominant hand mo is kanan, ang una mong tatanggalin, the first one you need to remove is the non-dominant left. Kung ang dominant mo naman ay kaliwete or kaliwete ka, ang una mong tatanggalin, nasa kanan. So you start by pinching and holding the outside area near the wrist. So ipipinch mo class yung glove doon sa wrist area. Once done, ipipil mo siya in a downward motion away from the wrist. Then once done, natanggal mo na siya, napil mo na, you now enclose it. You pull the glove away and you enclose it in your glove hand. In your glove hand. I-close mo siya, class. Make sure that it is properly enclosed. Pasok na pasok yung natanggal na glove dun sa glove hand mo. Once done, you now go to the next step. Using your unglove, yung kamay mo na walang gloves, you get 2 fingers, you insert the 2 fingers under the glove. Now, be very careful here, class. Do not touch the outer surface of the glove. So, pag na-insert mo na yung dalawang daliri, you now pull it downward, class. You now pull it downward and you continue to pull it until ma-fully remove siya. So that's how you would remove or your doffing of your gloves. Which again, ituturo ko sa inyo when we meet in person next week. So this is the reference. So that ends activity 1 class.